Usually when we talk about getting a medical diagnosis, there's this like expectation of absolute precision.
Oh, definitely.
Like you break your arm, you go in, the x-ray shows that jagged white line on the film, and the doctor just points at it and says, there it is.
Yeah, it is a very comforting binary. Like the bone is broken or it isn't. The path forward is just obvious.
Right. But step into the world of personalized cancer medicine, and suddenly that x-ray machine is kind of useless.
Yeah.
We're looking at a diagnostic landscape that is just completely murky.
Absolutely murky.
So when an oncologist uses a machine learning model to, say, pick a cancer drug for a specific patient's tumor, and the model just spits out a recommendation like use drug A, how do we actually know the algorithm isn't just, hallucinating?
Well, the scary part is we often don't. And that is the terrifying reality of current point predictions.
A point prediction.
Yeah, so a standard machine learning model gives you a single guess based on the genetic makeup of the cancer cells. And I mean, in a low-stakes scenario like a streaming service predicting what movie you want to watch next, a single guess is perfectly fine.
Right. If Netflix is wrong, I just watch something else.
Exactly. But in a life-or-death healthcare scenario, a single guess is completely unacceptable. We really need to know the mathematical margin of error. If an algorithm predicts a chemotherapy drug will work, the doctor needs a mathematical guarantee of its reliability, not just a guess.
Okay, let's unpack this. Because reading through this paper by Len Hoff and the team, it feels like they are tearing down the entire foundation of how we currently make these predictions.
They really are.
Like before we even get to the artificial intelligence part, they argued the underlying data we feed these models is deeply flawed.
Yeah, and it goes back to the oldest rule in data science, right? Train an algorithm on bad measurements, you get bad predictions.
Garbage in, garbage out.
Exactly. So in cancer research, the traditional ways to measure how well a drug works are these metrics called IC50 and AUC.
Let's break those down.
Sure. So IC50 stands for the half maximal inhibitory concentration. Basically, it measures exactly how much of a drug you need to kill 50% of the cancer cells in a test tube.
And the other one? AUC.
Right. AUC stands for area under the curve, which integrates the drug's effect over a range of different doses.
Wait, okay, if I'm a researcher designing a treatment plan, why is IC50 considered a bad measurement? I mean, killing half the cancer cells seems like a very solid, objective benchmark to track.
Well, it is a solid benchmark if you are comparing how two different types of cancer react to the exact same drug.
Okay.
But the moment you try to compare the IC50 of drug A against the IC50 of drug B to see which is better for a single patient, the logic completely collapses.
Because of the toxicity, right. I was thinking about this using a visual analogy, like trying to compare the speed of a car and a boat to see which gets you to work faster.
Oh, that's a good way to look at it.
Right. The environments, or in the case of the drugs, the therapeutic concentration ranges are totally different. You can't just compare top speeds.
Exactly. The human body is the environment here, and it has very strict limits. A lower IC50 for drug A might look fantastic on a spreadsheet.
Meaning it kills cancer rapidly.
Right. Kills it rapidly in a petri dish at a very low dose. But to achieve that specific drug concentration safely in a human bloodstream, it might actually be impossible without killing the patient.
Oh, wow.
Yeah. Meanwhile, drug B might have a higher IC50 in the lab, meaning it takes more of it to kill the cancer, but it might be incredibly well tolerated by the human body at high doses.
So the test tube doesn't care if the patient survives the toxicity, it just cares if the cell dies.
Exactly.
So the established measures are practically useless for ranking different drugs against each other for a real human being. That explains why the researchers pivoted to this new metric they call CMax viability.
Yeah, CMax viability really changes everything. CMax represents the peak plasma concentration of a drug.
Peak plasma concentration.
Yeah, meaning after you administer the highest clinically recommended, legally safe dose to a human patient. CMax is the absolute maximum amount of the drug you can safely have in your blood before the side effects become life-threatening.
Okay, so how does that translate into a score an algorithm can actually use?
So imagine a graph. The researchers take the standard dose-response curve, that's the line showing cancer cell death, as the drug dose increases on the x-axis.
Got the graph in my head.
Then they draw a vertical line straight up from that safe CMax concentration point on the x-axis. The exact spot where that vertical line intersects the survival curve is the CMax viability score.
And that's a number.
Yes, it ranges from 0 to 1.
Meaning 0 is complete cancer cell death at a safe human dose.
Right, and 1 means the cancer is completely unaffected at that maximum safe dose.
So Think about it from the patient's perspective. Imagine looking at your own lab results. You wouldn't care what a drug does at a hypothetical lethal dose in a plastic dish.
No, of course not.
You would want a list of drugs ranked purely by what they can safely do inside your body. It standardizes the playing field entirely.
It really grounds the machine learning data in clinical reality. You are directly telling the predictive algorithm to only prioritize drugs that actually work at the doses doctors are legally allowed to prescribe.
All right, so time for a quick recap before we move on. Traditional metrics like IC50 fail at comparing different drugs because they ignore human toxicity. CMax viability fixes this by standardizing the measurement, basically calculating cancer survival at the maximum safe clinical dose.
Perfectly summarized.
So to you, the listener, think about it. If you were designing a treatment plan, wouldn't you want your data rooted in human clinical limits rather than theoretical lab
It
numbers?
just makes so much more sense.
But having a fair metric like CMax is just the foundation, right?
Right.
We still have the problem of the AI throwing out blind guesses.
Yeah, the point prediction problem.
Exactly. And this is where the paper introduces the core mathematical framework. Conformal prediction, or CP. I was trying to visualize this too. If a standard point prediction is like throwing a single dart blindfolded and just hoping you hit the bullseye.
Then CP feels like throwing a massive net over the dartboard, right?
Yes. Throwing a net and having a mathematical proof that the bullseye is definitely trapped inside that net somewhere.
That's a really great analogy. The whole shift here is moving from single answers to what they call valid prediction sets.
Valid prediction sets.
Right. So instead of pointing at a single class and saying the answer is class A, conformal prediction says, I guarantee the answer is either class A or class B.
Or a range, right?
Yeah. If we are predicting a continuous number, like our CMax viability score from 0 to 1, it provides an interval. It guarantees the true effectiveness score falls somewhere between, say, 0.1 and 0.3.
I grasp the concept of the net, but how does the math actually guarantee the bullseye is inside it? I mean, machine learning models aren't magic. They make mistakes constantly. How do you force a computer to basically admit it's unsure?
Well, conformal prediction forces the model to learn its own mistake patterns.
Oh, so
Normally in machine learning, you split your data into two parts, right? A training set to teach the model and a test set to evaluate it. But CP requires a third, totally distinct piece of data.
called the calibration set.
Exactly, the calibration set.
Wait, so the model trains on the first set.
Yeah.
Then what? Does it just like grade itself on the calibration set?
It runs predictions on the calibration set, but since we already know the true answers for this specific data, the algorithm calculates the discrepancy between its prediction and reality.
Oh, I see.
It builds a score distribution of its own errors. It maps out exactly how often it gets confused and by how much.
And then the user, like the doctor or the researcher, gets to set the boundary. The paper talks about setting a maximum allowed error rate, which they symbolize as the Greek letter alpha.
Right. You define alpha based on your risk tolerance.
So give me an example.
If you are an oncologist and you set alpha to 10%, you are mathematically demanding 90% certainty.
Okay.
The system takes that 10%, looks at the score distribution of errors from the calibration set, and calculates a precise threshold. When you finally run the model on new, unseen patient data, it widens the net of its prediction just enough to satisfy that exact threshold.
What's fascinating here is that conformal prediction isn't a brand new artificial intelligence algorithm that replaces everything else, is it?
No, not at all. It's a layer.
It sits on top.
Yeah, you take whatever predictive engine you've already built, plug it into this Python pipeline, and it forces that fuzzy algorithm to output a rigorous, statistically validated safety net.
The proplan they built is incredibly flexible. Like if your underlying model can provide some notion of uncertainty, CP can just convert that into a hard guarantee.
Exactly.
Okay, time to pause for a second. We've established that CP shifts ML from making single guesses to providing mathematically guaranteed ranges or sets. And it does this using a calibration data set and a user-defined error rate, like our 10% alpha.
Right, which locks in that 90% confidence.
So listener, how much more comfortable would you feel taking a harsh chemotherapy treatment if the computer didn't just boldly say drug A, but instead said, I am 90% mathematically certain the best drug is either A or B?
It definitely changes the conversation in the clinic.
It really does. But that raises a big question for me, though. If CP is just the net, what is actually throwing the dart in this paper?
Ah, the engine.
Yeah, what is the specific machine learning model doing the heavy lifting underneath it all?
So the researchers developed an algorithm specifically for this, and they called it ASARON-RF.
Sounds like something from Lord of the Rings. What does it stand for?
It stands for simultaneous regression and classification random forests.
Okay, hold on. Simultaneous regression and classification. Why do they need a new algorithm to do both at once? Why not just use two separate models, like one to classify if the drug works, yes or no, and another to predict the numerical score?
Because doing them simultaneously allows the model to share feature learning.
Share feature learning. Meaning what exactly?
Well, a random forest is a collection of many different decision trees. Think of them as like individual algorithms voting on an outcome.
Okay, a forest of voting trees.
By forcing the trees to predict the binary classification, yes or no, is it effective, and D, the continuous regression score, exactly how effective is it, at the very same time, the model gains a much deeper understanding of the underlying biology.
I have to push back here for a second, though.
Sure.
A random forest is just a giant collection of trees voting, right? Each tree votes yes or no. How does a simple binary voting system give conformal prediction the nuanced probability scores it needs to build those error distributions we just talked about?
That's the clever part. The probability comes from the spread of the votes.
The spread.
Yeah, so if 99 out of 100 decision trees vote that a drug is effective, the model has a very high built-in certainty, right?
Right. 99 to 1 is a landslide.
But if it's a 51 to 49 split, it's highly uncertain. That proportion of trees voting actually becomes the raw uncertainty measure.
Okay, so we have the raw votes, but the paper spends a huge amount of time explaining how you actually score that uncertainty to create the CPNet.
Oh, yeah. They went deep on the scoring rules.
They didn't just find one scoring rule and stick with it, right? They went through an entire evolution of failures and fixes to get this right.
They had to, honestly, because medical data is just incredibly complex. So they started by testing something called the true class score, or TC.
How does that work?
It simply looks at the probability the model assigned to the actual correct answer. So if a drug was effective, but the trees only gave it a 10% chance of being effective, the true class score flags that as a massive error.
Seems straightforward enough. Why didn't they just stop there?
Well, true class scoring often struggles to provide tight, useful prediction sets when the model is genuinely confused between multiple classes. It can be kind of brittle in practice.
Brittle, so they needed something more flexible.
Exactly. So they moved on and tested the summation score.
Summation, so adding things up.
Right. This technique sorts all the class probabilities from highest to lowest, and it just keeps adding them up until it finally hits the true class.
Wait, but if you just keep adding up probabilities until you hit the right answer, doesn't your prediction set like your net get ridiculously huge?
It absolutely does.
Wow.
That's the flaw. Summation guarantees the true answer is in the net, but the net might cover the entire dartboard.
Right, which isn't helpful.
Not at all. A doctor doesn't want an AI to say, I am 90% sure the best drug is either A, B, C, D, E, F, or G. You have high coverage, but terrible efficiency.
Here's where it gets really interesting. To solve this efficiency problem, they had to confront the biggest demon in cancer data: class imbalance.
Oh, class imbalance is a nightmare.
Most drugs fail against most cancers, right?
Oh.
You have way more resistant cells in these data sets than sensitive ones.
Yeah, and class imbalance just destroys standard AI models. Think about it. If 90% of the samples in your dataset are resistant, a lazy AI can just guess resistant every single time.
And it would technically be 90% accurate.
Exactly. It achieves 90% accuracy overall without actually learning a single thing about the biology.
But it completely misses the 10% of cases where the drug actually cures the patient, which entirely defeats the purpose of personalized medicine.
Right. So to prevent the minority-sensitive class from being statistically drowned out by the majority, they implemented the Mondrian score.
Mondrian, like the artist.
Exactly like the artist. Pete Mondrian, known for those grid-based paintings that compartmentalize space with bold lines and primary colors.
Wow, I can picture those, yeah.
So Mondrian conformal prediction fundamentally changes the calibration phase by compartmentalizing the data, just like those paintings. Instead of guaranteeing 90% certainty on average across the whole data set, it splits the calibration data by class.
So it forces the algorithm to prove it is 90% accurate on the resistant cases and separately prove it is 90% accurate on the sensitive cases.
Individually, yes.
Oh, wow.
It locks in the mathematical guarantee for the rare outcomes, ensuring we don't discard a potential cure just because it's, statistically inconvenient in the broader data set.
Okay, that perfectly handles the classification side, the yes or no voting. But what about the regression side? predicting the actual numerical CMax score.
For that, they extended Saran-RF with quantile regression.
Quantile regression.
Yeah, rather than just predicting a single mean value, quantile regression estimates the dispersion.
Dispersion meaning the spread.
Right, the spread of possible response values based on the data. That estimated spread dictates exactly how wide the numerical interval needs to be to guarantee the true CMax score falls inside it 90% of the time.
All right, another quick recap. Saran-RF is our engine, a forest of decision trees that simultaneously classifies and scores drug sensitivity. And CP uses specific scoring rules, specifically the Mondrian score, to compartmentalize the data and lock in that certainty, even for unbalanced rare edge cases.
That's the mechanism in a nutshell.
So listener, think about your own experiences looking at data. Do you often find that minority edge cases get drowned out by the majority? How crucial is a tool like Mondrian scoring to protect those minority outcomes? It's literally a lifesaver here.
It really is the difference between finding a cure and missing it entirely.
Okay, but this theoretical math is airtight in a simulation. Cancer data is notoriously noisy in the real world. What happens when this pipeline hits actual patient data?
Well, the researchers unleashed this on the GDSC dataset.
GDSC?
The genomics of drug sensitivity in cancer. It is a massive real-world database.
And they ran a specific case study, right? Comparing cell line 55 and cell line 13.
Yeah, they walked through this brilliant example using those cell lines against two drugs, 5-fluorouracil and vorinostat.
Okay, let's look at cell line 55. The sRNA-RF engine runs, the Mondrian CP scoring applies its net, and the output for 5-fluorouracil is the set containing only the number 1, just the 1.
Right, where 1 means sensitive. Because the conformal prediction set only contains a single element, and it's backed by that 90% confidence threshold we set earlier, the system is unequivocally stating this drug will work on this specific cancer.
And for vorinostat on that same cell line, the output was just 0 resistant. So it's highly confident it will fail.
Exactly. And once the classification identifies 5-fluorouracil as effective, the regression side takes over to tell us how effective.
And what did it predict?
It predicted a CMax viability interval of 0.1 to 0.3.
Keep in mind, 0 is complete cell death.
Right. That interval means it's predicting an incredibly lethal response to the cancer at a totally safe human dosage.
Those individual case studies are super compelling, but looking at the macro data across the entire GDSC dataset, the reduction in false positives is what really caught my attention.
Oh, the false positive reduction is massive.
False positives are the nightmare scenario in medicine, right? And AI confidently telling a doctor to use a drug that will actually do absolutely nothing.
Yeah, waste time a patient doesn't have.
But by adding this conformal prediction framework, they eliminated 52% of the false positives.
Over half. It cuts the AI's hallucinations in half simply by forcing the system to mathematically prove its certainty before making a recommendation.
I have to admit, I'm looking at the precision numbers in the results and I'm a little skeptical. After filtering out the uncertain guesses, the prioritized drug lists hit a median precision of 92%.
92%, yeah.
Hold on, 92% precision in oncology data. That sounds absurdly high. What is the catch here?
The catch is that this 92% applies specifically to the prioritized list.
Meaning what?
It's the precision on the predictions, where the model successfully achieved that tight 90% certainty threshold with a small prediction set.
Oh, I see.
For the highly ambiguous cases where the biology is just unclear, the conformal prediction set remains large. It's essentially telling the doctor, the data is too noisy, we don't know. It doesn't pretend to be perfect everywhere. It's just 92% precise when it claims to be certain.
Okay, that makes a lot of sense. It admits when it doesn't know.
Exactly.
And the paper notes that 75% of the time, the prioritized list generated by the model contained the absolute most efficient drug for that specific cancer sample.
It is huge.
If you're an oncologist staring at a complex tumor profile, you aren't looking at a black box algorithmic guess anymore. You're looking at a prioritized list where the top drugs have a rigorous mathematical guarantee backing their effectiveness.
If we connect this to the bigger picture, it transforms the AI from a dangerous suggestion box into an actual, reliable medical instrument. Right.
It establishes the one currency artificial intelligence currently lacks in healthcare, which is trust. We can finally trust the margin of error.
Okay, let's wrap this up with a final recap. Applied to real GDSC data, the combination of CMax, Saran-RF, and CP successfully weeded out over half of false positives and delivered highly precise, prioritized drug lists for specific cancer cells.
Right.
So, listener, If you were a doctor, how much time would a 92% precise, prioritized list of treatments save you in a critical moment? We journeyed from flawed Petri dish metrics to the clinical reality of CMax viability. We saw how the Saron-RF engine generates probabilities from a forest of decision trees, and how the Mondrian conformal prediction framework forces those probabilities into mathematical guarantees that protect the rarest, most vital outcomes.
Yeah, but there is a detail nestled in the discussion section of the paper that points to where this actually has to go next.
Oh, where's that?
This entire model, as impressive as it is trained on monotherapies.
Single drugs. Right, single drugs. But cancer is notoriously adaptive. It mutates. It develops resistance to single drugs rapidly.
Which is exactly why combination therapies, using multiple drugs simultaneously to trap the cancer from different biological angles, are the future of treatment.
Exactly. The authors acknowledge that moving from predicting the effect of one drug to predicting the synergistic effect of drug combinations is the vital next frontier for this technology.
Wow. Think about the sheer scale of that problem. We've built a mathematical net that works brilliantly for one drug, but the potential combinations of multi-drug therapies are near infinite.
It's astronomically complex.
Imagine looking at millions of possible chemical combinations. So, listener, think about this. If conformal prediction can mathematically guarantee the effects of a single treatment, how might this exact same framework be used to navigate the near-infinite combinations of multi-drug therapies?
It's a fascinating challenge.
Could this mathematical net be the key to perfectly cornering highly adaptive, mutating diseases with 90% certainty? That is something for you to mull over until our next deep dive.